My name is Marie Skłodowska. I work here at the University of Seville. For the last 15 years, I've devoted my research trying to understand how brain tumors work. It was not love at first sight. I've been working on different neurological diseases. In all of them, neuroinflammation is always there. And that's exactly what we are at the moment. Try to understand the impact of neuroinflammation in the onset and progression of brain tumors.
What exciting advances in cancer research are you involved with?
Yes, of course. I guess two of the main sites we have to work on in brain tumors is to get earlier detection and better therapies. On one side, I've been working in collaboration with the University of Oxford trying to catch micrometastases in the brain, which are like 10 times smaller than the metastases that we can detect by usual techniques. And on the other side, we are trying to work on the power of immunotherapy. If we analyze brain tumors carefully, it's made up not just by cancer cells, but other immune system cells. And that's exactly what we are trying to do, to awake the immune response, particularly microglia and macrophages, and become those cells more anti-tumoral.
How do these advances impact cancer patients today?
Well, today it's we have to be careful with these things because it's not going to be easy, it's not going to be soon, but we have really hope that the immunotherapy can definitely impact on the expectancy, life expectancy of brain tumor patients. These days are very, very poor. The life expectancy is measured just in months. So if we are able to make the immune system cells that are kind of suppressed at the presence of tumor cells and making it more pro-inflammatory, more anti-tumoral, we actually believe that in combination with other gold standard therapies like chemo or radiotherapy, we can actually, if not destroy all tumor cells, become chronic this disease.
What is your message of hope to cancer patients?
Well, there are many of them trying to work on this awful disease. We're gonna get it. Maybe it's not gonna be tomorrow, but I'm pretty sure that with time and with good investment, we are in a good position to say that we'll be able to give better life expectancy to brain cancer patients.
I'm Rosetta Ladrón from University of Seville. I have a PhD in neuroscience. During my thesis, we discovered the interaction between neural progenitor cells and glial cells in a lesioned brain. We demonstrate that this interaction is through gap junction communication. So after my thesis, I got a postdoctoral position in Salamanca. In my group, they have been demonstrated the importance of the connexin 43, the protein responsible for gap junction in cancer. They have been demonstrated that this protein is an antitumoral protein. So in that moment, I decided it's inspiring me to study this protein in cancer.
What exciting advances in cancer research are you involved with?
As I mentioned before, during my postdoc, I began to study cancer, in concrete glioblastoma, that is the most aggressive brain tumor. We studied the effect of a peptide based on connexin 43 in glioblastoma. And we demonstrate that this peptide is very important because it inhibits the proliferation and the invasion of the glioma stem cells. And also we demonstrate that the peptide promote the survival of the mouse model with glioblastoma. I think that it is the most relevant result of my postdoc.
How do these advances impact cancer patients today?
We demonstrate that this peptide have an antitumoral property, but this peptide was also proved in a sample of patients, a patent-derived samples, and we demonstrate that the peptide inhibited the proliferation of this sample, and also inhibited the invasion of this sample. It is very important because we directly obtain the sample from the Hospital of Salamanca, and we proved our peptide directly. It was very, very important for us.
What is your message of hope to cancer patients?
Glioblastoma is the most aggressive brain tumor. There are a lot of research looking for the cure for this tumor. In the last year, they have a significant improvement in the treatment of other kind of cancer. So I think that soon we are found a new treatment for glioblastoma.
Hi, my name is John Rowell. I am the Strategic Director at IRCAN, the Institute for Research of Cancer and Aging in Nice. And in particular, my focus is on multidisciplinary collaboration between researchers and innovation.
What exciting advances in cancer research are you involved with?
Now, cancer is a very complicated disease. And in fact, the way that I always look at it is it's like the universe. It's made up of many, many different galaxies, and each one of these galaxies is unique, having a different number of stars, planets, moons, asteroid belts that works. And this is how an individual cancer is. Each individual cancer is unique, composed of a multitude of different aspects. Now, one of the big challenges we're facing in cancer research is how we can understand the interplay of all the different components contributing to an individual's unique cancer. And this covers all the aspects of biological research, from basic biochemistry all the way up to cells, tissues, microenvironments, how the cancer is fitting into the tissue and the organism as a whole. And this is a monstrous amount of work. It's a huge amount of complexity in these cancers, but we are matching that in research. Cancer research is really going towards a huge amount of collaborative efforts. There are many, many networks within Europe, within the world, working together, bringing researchers together to try and understand what is happening. And the more we understand it, and the more that we can actually build on all the knowledge we have, the easier it is for us to identify new ways of treating it, a new understanding of how it develops, a new way of preventing it or screening for it. And that, for me, is the reason why I find cancer research so engaging. I think it's one of the few examples out there where you're going to bring together all different types of researchers looking at different aspects together to solve this common problem, a problem which affects the world as a whole. In IRCAN and in Ulysseus as a whole, we are working together to try and not only just understand cancer and the complexities, but to find possible solutions. And what's really important is how we can translate our basic research knowledge into possible clinical application. This is actually called translational research, and this is a big component of what we're doing.
What is your message of hope to cancer patients?
And I think the biggest output, the biggest thing that patients need to take to heart is every single day, week, month, year, we know more. Every single week, day, month, year, we're actually finding new opportunities, new methods, refining, optimizing, and finding new innovative solutions to help beat cancer. So it's getting better, and every day it's going to get better.
Hi, I am Jean Wang. I'm a scientist here at the Institute for Research on Cancer and Aging in Nice, where I am working on developing new ways to treat cancer. I became interested in cancer research about 12 years ago when I joined the Institute as a master's student. At that time, I realized that when the cancer cells are obviously the bad guys, they are getting the help of the normal cells that surround them. Since then, I am trying to understand how the normal cells can help the cancer cells to grow so we can come up with more specific strategy to treat cancer.
What exciting advances in cancer research are you involved with?
At the lab, we are working on metastatic breast cancer. The good news for patients is that for this type of cancer, if it is detected early on, the chances of getting cured are really high thanks to surgery and chemotherapy. However, the real problem begins when cancer cells already escape the primary tumor, here the breast, so they would colonize another organ such as the lung, and they would form what we call lung metastasis. The problem with this lung metastasis is that they are usually resistant to treatment, and here at the lab we are trying to understand why these lung metastases are resistant to chemotherapy. Using different methods, we were able to show that after a chemotherapeutic treatment, a lot of white blood cells are actually coming into the lung to surround this lung metastasis. This was very surprising because usually white blood cells are here to protect us against pathogens such as bacteria and viruses. We were able to show that here, these white blood cells that we call neutrophils are actually protecting the cancer cells from chemotherapy. More importantly, we were able to show that neutrophils could protect the cancer cells from chemotherapy by releasing filaments of DNA, which are called neutrophil extracellular traps, or nets. You can think of it like Spider-Man, who is releasing his web to trap the bad guys. Here the neutrophils are doing the exact same thing. They are releasing webs of DNA to trap the cancer cells and protect them from chemotherapy. In this context, at the lab, we showed that by digesting these filaments of DNA, we were able to resensitize the cancer cells to chemotherapy.
What is your message of hope to cancer patients?
So the good news for patients is that there is already in the clinic a medicine that can be used to digest these filaments of DNA from these neutrophil extracellular traps, a medicine called DNase-1. Indeed, DNase-1 is used in the clinic today as an aerosol to target the lung of patients with cystic fibrosis. And we do think that this medicine could benefit thousands of patients with lung metastases because it could resensitize the cancer cells to chemotherapy by digesting the filaments of DNA released by the neutrophils. At the lab, we do think that by understanding how the cancer cells interact with their environment, we can come up with more efficient way to treat cancer. In this context, more and more laboratories over the world are working on strategies to target both the cancer cells and the environment so we can achieve better results. I am confident that in the near future, more and more strategies will be used in the clinic on a daily basis to improve cancer treatment and to improve the quality of life of patients.
Del Mastro. I am a professor of oncology at the University of Genova, and my main field of research is breast cancer. I am involved in both translational and clinical research on breast cancer.
What exciting advances in cancer research are you involved with?
The most important results of the academic research carried out by my team include the identification of a new strategy to preserve the fertility of young breast cancer patients candidates for adjuvant chemotherapy. This strategy is now included in main national and international guidelines as a strategy to be proposed to all young breast cancer patients who want to preserve their ovarian function. Another important result of our research included the identification of a new, less toxic and more effective adjuvant chemotherapy regimen. This regimen is considered by national and international guidelines as one of the most effective treatments to be proposed to high-risk breast cancer patients candidate for adjuvant chemotherapy. And another important result is the identification of a more effective adjuvant endocrine therapy for postmenopausal breast cancer patients with the hormone receptor positive breast cancer candidate for adjuvant endocrine therapy.
What is your message of hope to cancer patients?
The most promising things in the near future in oncology, in my opinion, are the availability of new, most effective drugs, such as the antibody-drug conjugates, and the possibility to treat in a personalized way the patients by targeting the specific biological characteristics of the tumor. We expect a real revolution in the treatment of tumor or cancer in the near future.
I'm Marco Tagliamento. I am a medical oncologist and assistant professor at the University of Genoa. I am a lung cancer specialist and I am dedicated to clinical and translational research in the field of thoracic cancers. So in the last years, I've focused my research on molecular medicine and particularly on a tool that is increasingly being used in oncology, that is liquid biopsy.
What exciting advances in cancer research are you involved with?
Liquid biopsy is a non-invasive method to study the tumor by the means of a blood test. So particularly, we can study the DNA coming from different types of cells and released in the blood. And currently, we are working on defining the role of some specific DNA mutation that may affect white blood cells. And this phenomenon is called clonal hematopoiesis. So as you know, white blood cells are part of the immune system, and the immune system is crucial for anti-cancer response. So we are trying to deep how this mutation may affect the efficacy of anti-cancer therapies in patients with solid tumors.
How do these advances impact cancer patients today?
The results of this research program will have a positive impact since they can help to better stratify the probability of success of some treatment, and particularly the treatment that have an immunomodulatory effect, such as immunotherapy that is currently being used for most types of solid tumors.
What is your message of hope to cancer patients?
So our message to patients is to believe in research, because it is research that brings new opportunities and drives the progress in oncology.
My name is Raphael Heist. I'm a professor at the MSCA in Innsbruck. And one of the main topics I'm interested in is the topic of food systems. So food systems are very important for cancer prevention, because we know obesity rates are increasing in Europe. We know that in Europe, for example, the overweight and obesity rate is approximately 60% now. And this is by many considered a major warning sign.
How do these advances impact cancer patients today?
Well, there is more and more evidence which clearly shows that our diet is related to our metabolic health. And we also know that some aspects of our diets are worse than others. For example, the consumption of ultra-processed food, usually high in added sugar, in fat, in salt, and also chemicals, that they may have adverse effects on our metabolic health. So we have to think about, well, what to do with that kind of knowledge and how to apply it to practice. For example, how can we improve school food environments? How can we implement targeted taxes? And how can we increase the transparency for consumers in their daily food choices?
What is your message of hope to cancer patients?
Well, the good message is that our food systems actually can be fixed. We can introduce smart regulations which reduce the dominance of ultra-processed food. We can incentivize people to make wiser and healthier choices. And we can also incentivize the industry to change their recipes to offer healthier food. And I think it's not only about our health and about preventing cancer. Changing the food systems is also about our environment, about climate, and is a great opportunity to make the world a better place.
I'm a cancer researcher at the University of Montenegro, its Institute for Advanced Studies, and head of Molecular Medicine Lab at Faculty of Medicine at the same university. What inspired me to pursue a career in cancer research is the remarkable complexity of cancer cells biology and their ability to evade many different treatment strategies. Specifically, during my PhD studies, we found that a simple change of carbon source in the growth medium of the cells had a profound effect on how these cells responded to certain stressors, which then led me to the field of cancer metabolic reprogramming.
What exciting advances in cancer research are you involved with?
My current research is more of a translational nature, and I work on identifying new biomarkers for early detection of cancer, specifically colorectal cancer. My focus is on the role of microRNA, which are small non-coding RNA molecules important for the regulation of gene expression. They are frequently deregulated in many cancers and have a role in many biological processes implicated in tumorigenesis and can have either pro or oncogenic or anti-oncogenic role. MicroRNA are part of so-called liquid biopsies, which in my opinion represent one of the most exciting recent advancements in cancer research, because not only that liquid biopsies enable us to do a non-invasive detection of cancer at a very early stage before clinical symptoms appear, they also offer a tool for real-time monitoring of cancer progression and response to treatment. Combining newly identified biomarkers with existing diagnostic strategies is aimed at improving diagnostic efficacy and to improve detection of cancer at an earlier, more treatable stage. This will ultimately lead to improved patient management, to improved survival, decreased chances of complications, and in some cases, increased even chances for curative treatment.
What is your message of hope to cancer patients?
My message to cancer patients and their families is not to be discouraged with the diagnosis and to fight back, to be hopeful that the advancements in cancer research being made every day by many excellent teams of cancer researchers throughout the world who work devotedly to try and better understand this disease in order to fight it ever more effectively. In particular, advancements made in detection of cancer at the earliest stage are of particular importance because it is precisely at that stage when the chances for successful treatment are highest.